Hello everyone, I am Shubham Yadav from Patil Institute of Pharmacy, Talegaon Shamji Panta, Varda, and I welcome you all in Student Presentation Week 2025. I am in B Pharmacy, 5th semester, subject Pharmacognosy and Phytochemistry 2, and my topic is Pharmacognostic Study of Cinnamon Bark. In this topic, we will cover introduction about cinnamon bark, common name in various language, synonyms and taxonomy, biological classification, macroscopic characteristics, microscopic characteristics, chemical constituent, chemical test, quality control, method of preparation, marketed preparation, application of cinnamon bark. Introduction. Cinnamon bark is a spice derived from inner bark of cinnamon tree. Its distinctive flavor and aroma make it a popular ingredient in various cuisines worldwide. Beyond culinary uses, cinnamon bark is removed from its medicinal properties, traditionally utilized in herbal medicines for centuries. This presentation explores its introduction, preparation, and diverse application. Common names in various languages. Cinnamon bark is known by various names globally. In Spanish, it is referred as canela, while in French, it is called canela. Understanding these common names enhance its recognition and acceptability across different cultures. Synonyms and taxonomy. Synonyms for cinnamon bark include dalchini, Ceylon cinnamon, Cortex cinnamoni, Chinese acacia, and variation like Sichuan cinnamon. Many synonyms refer to the specific species or types of cinnamon, such as Cinnamomum zeylanicum, true cinnamon, and Cinnamomum cassia, Chinese acacia, which are also terms used to describe the bark. Biological classification: Kingdom Plantae, Division Magnoliophyta, Class Magnoliopsida, Order Laurel, Family Lauraceae, Genus Cinnamomum, Species Cinnamomum verum, Cinnamomum cassia, Cinnamomum laurerea, Cinnamomum burmani. Macroscopic characteristics: Form and shape. The bark is generally found in the form of rolled peas known as compound quills, which are made of single and double quill rolled together. Color: the outer surface is dull yellowish brown, while the inner surface is darker yellowish brown. Odor: cinnamon bark possesses a distinctly pregnant and aromatic odor. Taste: it has an aromatic and sweet taste in warm sensation. Size: individual quills can reach up to 1 meter in length with diameter of 1 centimeter and thickness of approximately 0.5 mm. Surface features: the outer surface is smooth but features faint scars from the position of leaves and buds as well as longitudinal striation. Fractures: the bark is brittle and breaks with a splintery, fibrous fracture. Microscopic characteristics: stone cells, scleroids. These are thick-walled lignified cells that provide structural supports. In some species, like Cinnamomum zeylanicum or true cinnamon, they may be horseshoe-shaped with U-shaped lignification. In other species, such as Cinnamomum cassia, they are often described as rectangular and thick. They can be found in group as scattered throughout the brake. Fibers. These are elongated, thick-walled cells that also contribute to the bark's fibrous texture. They are often found in groups and may have pointed ends. Oil cells. These are large, specialized cells that contain the volatile oil responsible for cinnamon's characteristics, aroma, and flavor. They appear as isolated cell or associated with other tissue. The distribution of these oil cells can vary with the age of the bark. Starch grain. Small, simple, and compound starch grain are found within the parenchyma cell of the cortex, secondary phloem, and medullary rays. Calcium oxalate crystals. These crystals, often needle-shaped, acicular rapids, can be found within the parenchyma cell, particularly in the medullary rays and phloem. parenchyma cell. These are the primary unspecialized cell that made up of bulk of the bark tissue. They store starch and other substances. Chemical constituent. Cinnamon contain about 10% of volatile oil, tannin, mucilage, calcium oxalate, and sugar. Volatile oil contain 50 to 65% cinnamon aldehyde along with 5 to 10% eugenol, terpene hydrocarbons, and small quantities of ketones and alcohols. Chemical test. First, a drop of volatile oil is dissolved in 5 ml of alcohol and to it a drop of ferric chloride is added. A pale green color is produced. Cinnamonic aldehyde gives brown color with ferric chloride whereas eugenol gives blue color. Second, the alcoholic extract is treated with phenylhydrazine hydrochloride. It produces red color due to the formation of phenylhydrazone of cinnamonic aldehyde. Quality control. To ensure the quality of cinnamon bark, rigorous quality control measures are implemented. This includes testing of moisture content, essential oil concentration, and the presence of contaminants. Standards set by the Food Safety Authority guide those practices, ensuring the consumers receives a pure and effective products. These measures help maintain the spice's integrity and extended its shelf life. Bark structure. Pure cinnamon, or also called as true cinnamon, is very thin, can be rolled into quills, and has a smooth inner surface. Cassia, a common adulterant, is thicker and contains multiple rough layers. Aroma and taste. Good quality cinnamon has a warm, sweet, and woody aroma and slightly sweet, warm taste. Poor quality cinnamon may be bitter, metallic, or have chemical scent from adulterants. Color and moisture. The outer surface of cinnamon is dark red brown, while the inner surface is red brown and smooth. It should not be wet, as high moisture content can lead to mold. Method of preparation. First, harvest the inner bark. Second, dry the bark. Third, grinding. Fourth, soxolate method. First, harvest the inner bark. Cut young, thin stem at a precise angle after a rainy season, then carefully peel the outermost bark layer before stripping the tender inner bark into the sheets. Dry the bark. Carefully remove the outer bark and inner bark from the branch. Then arrange the thin natural bark strips on the surface, such as a rope or mesh, in well ventilated area from direction. Ensure good flow of air and a warm environment as this helps the strips curl into quills and dry completely in a few hours to several days. Third, grinding. First, snap cinnamon sticks into smaller pieces to make them easier to process. You can then use a higher speed blender on a dedicated spice or coffee grinder, running it in short bursts for best result. Traditional method used as a mortar and pestle. Soxhlet method. The Soxhlet method for cinnamon bark involves placing ground cinnamon bark into a thimble and using continuous refluxing soxhlet such as hexane, ethane, or petroleum ether to extract cinnamon oil. Marketed preparation. It is one of ingredients of preparation known as Rumalia gel, Coughlette lozenge, Chyawanprash, which is from Himalayan Drug Company, Garbhapal Ras, Toothsekhar Ras, Dabur, and Sage Staminax capsule, stage herbals. Application of cinnamon bark. Antioxidant. Rich in antioxidants, cinnamon bark help reduce oxidative stress, which can damage cell and contribute to chronic disease. Anti-inflammatory. It exhibits anti-inflammatory property due to its polyphenol which can help manage inflammation in the body. Antimicrobial. Cinnamon is effective against various bacteria, fungi, and viruses, potentially acting as a natural preservative and helping to prevent infection. Antiviral. It contains compound that shows antiviral activity contributing to its use as a natural remedy against infection. Antifungal. Cinnamon bark can help fight fungal infections such as those caused by Candida or Athlete's foods. Acknowledgement. I express my heartfelt gratitude to Dr. Nilesh Banarse sir, our estimated guidance for his continuous support, motivation and valuable guidance. I am thankful to respect Principal Sir, Director Sir, all the faculty member and non-teaching staff for their encouragement and assistance. Special thanks to my friends and classmates for their cooperation and moral support. So here our topic has finished and I hope you will like this information. Thank you.